I'm Jeff Gray. I'm associate professor here at Johns Hopkins, and my major area of research is in protein structure prediction and design. I work on protein interactions and protein interactions with surfaces, like in biomineralization processes, also antibodies, and a little bit of protein design. What I want to talk about, well, this semester this is an elective class, so I want to have a little bit of fun and teach people basically the general approaches in protein structure prediction and design. So before we get too far, let me just ask, why do we want to engineer proteins? Why did you take this class? What do you want to learn in this class? Why do we want to engineer proteins? And I like to do some back and forth and participation in the class. I know it's a little hard when you're just getting to know each other. How about this? Let's just pair up and tell somebody next to you. See if you can come up with 3 or 4 reasons why you want to engineer proteins.
Yes. So I mean, proteins are really high functioning, so if you can they bind and react really specifically and quickly.
Yeah, proteins are high functioning. They bind and they react. What else? Krishna, did you guys come up with anything?
Yeah, we thought that if we engineer enzymes, maybe we can use them for biocatalysis.
Enzymes used for catalysis, biocatalysis. Yeah. Go ahead.
We can make sensors and localize them to various places.
Sensors. We can also maybe do localization, get things to go where we want them to go. Other things? Jelly Oscar? Sorry, I'm picking at all the people I know.
All right, so say you've got an epitope in your enzyme that is autoimmune, your immune system reacts against it, you might want to design it out and maintain function.
Sure, immunity engineering, say, get rid of an epitope in a vaccine or a drug or something that we want to get rid of. Yeah, these are all good reasons. So why are proteins such a good molecule for doing this? So, so proteins are the building block of the cell. It's what all the, well, not all, but it's what a lot of our DNA encodes for. So the big thing about proteins is we get a very particular structure. They fold into a structure and they are able to perform some kind of function, right? So they can perform a particular chemical or structural function, often because they have a particular structure. So one of the magic things about proteins is they fold and they have structure. Not all of them, but a lot of them. So that specific structure and the specific surface chemistry they present gives them particular properties and allows them to do things, from binding things to reacting to things to performing being particular structures, say acting in the cell. So those are great properties to be able to exploit. Okay, so what are proteins? Everybody knows this. What's a protein? It's a polymer. A polymer of? So proteins are polymers of amino acids. But a protein's not, so the polymers are made up of the naturally occurring 20 amino acids, and but they're not like any other polymer, right? Like, so I'm a chemical engineer, so proteins are polymers, but they're not exactly, right? How's a polymer different than a protein in particular? You know what I mean by polymer? It's like polyethylene or polystyrene. They make things out of polymers all the time.
It doesn't have any secondary structure, tertiary, quaternary.
Yeah, so proteins have structures. They might have secondary structure, helices and strands. So proteins are composed of amino acids. Polymers could be any particular, any repeating chemical group. So you could have different, say, ethylene glycol, vinyl chloride, something like that. But we can make polymers, right? We've been doing this since the 60s, and even before that we've been making polymers. We're very high-tech polymers. We do a lot of very particular things with them. But why do we want to engineer a protein? I mean, why are they better than polymers?
Proteins are specific?
They're to be very specific. So the proteins are going to have a specific amino acid sequence. And this could be basically any sequence of residues that we put together in the gene to code for the proteins. Whereas polymers, you're typically polymerizing things. You can have heteropolymers of different things, but they're just long repeats. Polymers would be often would be a mix of lengths, whereas a protein would have a particular length. So it's one of the very few molecules that we can exactly specify what we want the molecule to be and get up to very large molecular weights. So we have exquisite control over the chemical structure of the proteins. And all of this leads up to our central paradigm of protein structures. Is that the sequence codes for a structure, and the structure codes for a function. Right, so we can specify a particular sequence of a protein. That protein then will fold up into a particular structure. That particular structure will have a particular function. In evolution, the cells are evolving to optimize some kind of function or some kind of fitness, and often that means creating a function, evolving on the sequence level to make this function happen, and so there's a feedback loop between this. Okay, so in order for us to understand if we want to engineer proteins to bind stronger or do particular reactions or make a particular nanoshape, we want to be able to understand the sequence structure relationship and then how that structure then relates to function. So that's one of the big goals of the class, is to understand quantitatively how these pieces fit together. Okay, so if we want to do, so nature, well, if you observe nature and look at a genome sequence, we go in this direction. The sequence makes the structure, makes the function. But if we want to do design, we need to do the inverse of this. We need to take some function, decide from there what structure you might want to do that function. So say if you had a particular reaction you want to do, you need to put a nucleophile in the right place and a hydrogen bond donor in another place, and a place for the products to come out somewhere else, and you position these groups in space, so you might define a structure, and then from that structure you might then say, well, what kind of sequence could fold into that structure. So the design process is going to go in reverse. We're going to start with the function to derive the structure to derive the sequence. Right. So proteins, polymers, amino acids. Everybody knows what an amino acid is, right? We've seen this probably since high school. We have the amine is a nitrogen. Nitrogen will have a couple protons. It'll have an alpha carbon, so-called alpha carbon. It has a hydrogen on it and has some kind of functional group on the side. And then we'll have a carbonyl group, C double bonded to an oxygen. And in its single form as a single amino acid, that'll be an acid group. That'll have a COOH on it. All right, so that's our single amino acid. This is our amino group. This is our carbonyl group. And it's fairly simple, but it's what gives rise to our interesting chemistry, because we have both the nitrogen and the amino function, and we also have the acid function of the carbonyl. And then we have that R group that allows us to get a lot of chemical complexity. Okay, so there it's polymerized via a peptide bond. So if we take the nitrogen, the alpha carbon, and the CO, we can put a second one next to that with another nitrogen, an alpha carbon, and a CO. To do that, we need to take away a proton from the nitrogen and an OH from the carbonyl. So that's going to take away water, so this is going to be a condensation reaction. It produces water. We get rid of water to make the amino acid bond. So this is, there's the peptide bond. And let's see, we still have a hydrogen on the nitrogen. Now we have multiple side groups here, R1, R2. We can keep going. N, C, R3, CO. Still have a proton up here. Still have a hydrogen on the alpha carbon. And here's my amino acid group that repeats. R equals a side chain. There are 20 naturally occurring amino acids. And those are what make the interesting chemistry and make it do different things. So we have different proteins that fold into different things. It's because we have different sequences of side chains. Okay, so how many people know this stuff? You've seen this before? Okay, so how many people know there are 20 amino acids? Yeah? Okay, why don't we get into groups of 2 again, and why don't you see if you can write down, should we go for all 20? Or just do like 3 or 4? Why don't you see if you can make 3 or 4 and draw chemical structures? See if you can draw 3 or 4 chemical structures. We can work in pairs and see what you can do. So there's some major classes of amino acids, right? What are the major classes? So there's hydrophobic amino acids. What are the hydrophobic amino acids? Just for review, let's just spit them out because you guys know.
Glycine.
Valine. Leucine, isoleucine,
Alanine.
phenylalanine, alanine,
Proline.
huh?
Proline.
Proline. Okay. What else? What is besides hydrophobic? What else do we have?
Aromatic.
Yeah, let's do aromatics. Which ones are aromatic?
Tryptophan.
Tryptophan, tyrosine. When we are, these don't have to be exclusive categories, right?
Phenylalanine.
Okay, what else? How about charged amino acids?
Glutamine.
Glutamine, serine? Not serine. But you could make serine, how can you make serine charged? You can take a lot of pH. Maybe with kinase, we could use serine to a bit of kinase. Phosphorylated, that would make a lot of charge in your serine. So phosphoserine for sure. Glutamine, aspartic acid, glutamic acid. What's the charge on those?
Negative.
Okay, so negative. So somebody probably knows all of these questions in here. By next week, everybody should know the answer to all of these questions, at least to memorize it once. If you forget it later, that'd be okay. So those two are negative. How about positive charged amino acids?
Lysine.
Lysine. And one more. It's a pirate's favorite. Great. What else? Aromatics, hydrophobics, and how about polars, non-charged polar amino acids? People have seen that three-letter code before. ARG for arginine. People have seen the three-letter code before, so you need to know your three-letter codes and your one-letter codes. There's also one-letter codes because, well, the three letters are convenient. That's nice when you print things out because people can remember them, but the one letter is really convenient for compact sequences, so you want to memorize those too. All right, what else? So polar amino acids. What are we missing?
Cysteine.
Histidine.
Cysteine.
Cysteine. Serine. Add another CH2 group to serine. Serine. Okay, how many do we have so far? 6, 7, 8, 9, 10, 11, 12, 13, 14. Wow, we're missing a few.
Missing methionine.
Methionine. It's kind of hydrophobic. Methionine. Glycine. Yeah, where do we put glycine? I don't know. It could be hydrophobic or polar, I think. It's got a CH2 group. There's our 20 standard amino acids. Non-polar aliphatics. So glycine is the lack of a side chain. We just have a hydrogen. Alanine is our smallest simple thing with a side chain, CH3, with a heavy atom side chain. Valine looks like a V. Leucine is a little longer, looks like a V. Isoleucine is 4 carbons, but rearranged from leucine. Branches early instead of late. And methionine has a sulfur in it. Aromatics, phenyl, tyrosine, tryptophan, we got those okay. So tryptophan has 2 rings, and the nitrogen, tyrosine has the hydroxyl group. If I asked you to draw amino acid with a hydroxyl group, you should have a few choices for those. Phenylalanine is straight hydrophobic. Let's see the next set, polar, uncharged, serine, threonine, cysteine, proline. So these guys are calling proline polar. It's got a lot of CH groups, so it's kind of on the fence there. But every amino acid is polar, right? Because every amino acid has the amine group and the acid group. So every amino acid is polar, and then the side chains modulate that a little bit. Oh, asparagine and glutamine. Yeah, so asparagine and glutamine. I missed those. So those are like the glutamate and aspartate that except with the nitrogen instead of another OH there. What else? Positively charged. Lysines, arginines, and histidine can be positively charged if your pH is lower than 6.8, 6.4, something like that. In biological context, histidine is often charged. And negative groups with the COO, aspartate is the short one, and glutamate has 2 extra ones there. Right, so there's the whole bunch. I gave you on another view, kind of the stereochemical view, so you can, they're grouped a little differently. Great. All right, any questions about the amino acids? So you should be able to tell me who's charged, not charged, who's got oxygen or hydroxyl group, which ones have acid groups, which ones are positive, negative, all the general properties of amino acid. I should be able to ask you for the quiz in a couple weeks, and then we'll all have that in our brains for doing that. Okay. Let's do a couple more structural pieces. I'm going to do a little bit of nomenclature so we can talk about things. NCCO. So that's our backbone nitrogen, our backbone carbon, and oxygen in the carbonyl group. As mentioned, that carbon we call the alpha carbon. And as we then go out to our side chain, we're going to label each of these atoms with a letter from the Greek alphabet. So C alpha, C beta, C gamma, C delta, and epsilon, C zeta, and C zeta. I'm not Greek, as you can tell. C zeta, and eta 1, and N eta 2. What amino acid do I have here? Give you a hint. Positively charged. This is arginine. So lots of CH groups. Arginine has the most labels that you need. So we label these C alpha, C beta, C gamma, delta, epsilon, zeta, and eta. So with most amino acids, we won't need that many letters, but we have a nomenclature to talk about this. What else? We have some angles. We call this angle phi, and this angle psi. We connect to want to go this way. We connect to the next amino acid down. Call this angle omega. And the side chains, we're going to use chi to label these angles. This is chi 1, chi 2, chi 3, etc. 4. 5. We don't worry about chi 6 too much because these protons can spin around pretty freely. But if we want to place all these heavy atoms, we need to know at least those torsion angles. Backbone, these are freedom. So if we're doing structure prediction and design, we need to put this stuff in the computer. We need coordinates for the atoms. We need to put the atoms somewhere in space. So I need to know things about the geometry. So one thing I need to know is bond lengths. How far is it between the nitrogen and the alpha carbon? How far is it between the alpha carbon and the carbonyl carbon? How far is it between the alpha carbon and the beta carbon? So it turns out that bond lengths are practically constant. So we've crystallized many structures of proteins, looked at many of them, and pretty much the N to C alpha distance is pretty much always the same. So we can look that up in a table. Turns out that bond angles are practically constant. Except tau, which varies 2 to 3 degrees. Okay, so I didn't show you tau. So bond angles mean the angle between three successive atoms. So tau is the angle around the alpha carbon right here. This is tau. So you have an angle between the N, the C, and the C alpha, the N, and the next C alpha. So every 3 atoms you can make an angle out of those 3 points, and those are the bond angles. Those are also practically constant. So I write practically because they're not exactly constant. They vibrate. You're in some kind of energetic well. It's essentially a quadratic well, and these things vibrate. That's how we get IR spectra, by looking at these resonant vibrations between the atoms. But if we're going to predict the structure of proteins, well, so far most of the good methods use average constant values for this. There's definitely a question in the research whether we could get better by letting these values vary a little bit, but for the moment we'll keep those constant. Okay, so bond lengths and angles are tabulated in paper by Eng and Huber back in 1991. So he's been around for a little while, and this is one of the articles that I posted on the website for reference. You don't need to read the whole article, but the tables will be useful, so you have these lengths and angles. Great, so if everything's constant, what's the tough part? Well, the tough part then are what's called these torsion angles. So the torsion angles, omega is about 180 degrees. So omega is this bond between the carbonyl group and the nitrogen across this peptide bond. I'm sure you've talked about the chemistry of this. This carbonyl group has resonance structures that makes this double bond resonate with the nitrogen and the free electron pair there that essentially puts this in one set of p orbitals that puts this peptide bond all together in a plane. Right, so omega is typically fixed at 180 degrees. There's one exception to that. You know the exception? The exception is cis-proline. Proline, which circles back on itself and binds to the backbone. Because of that, interaction can be on the cis side, meaning instead of these being, actually I kind of drew this cis, these should be drawn, it's hard to draw on the board in 3D, but they should be trans, and if they flip over to cis, that's via cis-proline. So proline has cis-pro, where omega is about 0 degrees in about 25% of prolines. Okay, so given that, that means phi and psi are the source of the most significant variability. All right, so these phi and psi's are torsion angles. If we take 4 atoms, we're looking at this bond. What the torsion angle is where we spin around the bond, right? So that's the angle we're looking at. If we draw, let me draw this peptide bond again. If we draw C alpha, CONH. So what happens is because of this bond, these all become planar. And we end up having this set of atoms all in one plane, okay? And that C alpha connects to the next CO, which connects to the next N, H, and the next C alpha. And these atoms all end up being the same plane. So then we have two planes, and those planes can swivel back and forth using those angles. So here's, again, this is the phi angle. It's going to twist this plane around the alpha carbon. And then here's the psi angle. It's going to twist the second plane around the alpha carbon. So basically you have a set of planes that then make a chain, and you have all these planes that you can twist around as you go down the chain. Okay, so these are planar peptide units. Okay. Great, so we then could look at what we could then, thinking like maybe a computational scientist, maybe not like an observational biologist, but like a computational scientist, you could say, well, okay, if that's true, if those are the rules I give you, then I could create a bunch of protein structures just by moving phi and psi and flopping these things around, and I should be able to see what proteins are going to do. Yeah, so that's actually been done, and people have calculated this. Again, Linus Pauling was one of the first ones to try and calculate what these structures are when you twist these angles around, and then Ramachandran was the one who eventually looked at a lot of data once we had a lot of structures. And it turns out, so as you twist these around, the problem is at certain twists, what's going to happen is these atoms are going to bump into each other, and they're not going to work. Okay, you're not going to be able to put these angles together. So that's kind of one of the first lessons of protein folding, is we start to move these angles around and do things clash and they just don't fit together, even with just the backbone, regardless of the side chains. This paper by Mandel in the 70s, based on the same kind of calculations that Pauling started with. If you look at when does the oxygen atom of the N+1 residue interact with the carbon atom of the N residue, so when does this oxygen and this carbon come and bump into each other, you can calculate that happens when phi gets to a certain value and it excludes things that are over here and only allows things here. So people have looked at this using ideal spheres and put things together and said, hey, a lot of times stuff bounces into each other. So if you do that, and you plot phi versus psi, and you exclude all of these regions where things bump into each other, you end up with three big regions of space. This region down here around phi minus 60, psi minus 60, which you all know is alpha helices. And then this region up here that's beta sheets with positive psis and negative phis. This stuff over here has to do with bumping into the L amino acid, the chiral amino acid. And then here's actual data of main chain dihedrals from high resolution protein structures. This is from Richardson's Anatomy and Taxonomy of Protein Structure. Jane Richardson's one of the pioneers who did some of the very first computer models, wrote some of the first programs to display proteins on the computer, and then she spent hours and hours just looking at these beautiful structures and watching the patterns and taking the data and tabulating and discovering all of this stuff. So Jane's article is fantastic to read and gives you a lot of detail, but here you can see this alpha region and this beta region, and they're a little bit different because it turns out things are a little bit squishy and the bond angles can vary a little bit and the bond distances can vary a little bit, and the chemistry of the side chains also matter. So the experiment's a little different than the original calculation there. All right, this is a plot for glycine residues. What's different about this plot? Here's the original Ramachandran plot, which if you've seen before, that's probably what you've seen. This is the one for glycine. How's it different? Go ahead.
Glycine is a lot more flexible, so you see that it has 5-side angles that are included in areas of the plot.
You have different regions. You have points over here where you don't have points before. I'm looking for a word that starts with S to describe this plot. Symmetric. This one's symmetric. Why is glycine symmetric?
Because it has two hydrogen on the alpha carbon.
There's a hydrogen on the alpha carbon. So glycine and CHHCO. There's two hydrogens. There's no L or D form, whereas for other amino acids, it's always an L form. There's always a particular stereochemistry to this alpha carbon, and that stereochemistry breaks symmetry, and so you only have this asymmetric diagram. But here it's symmetric, so the left and right should be the same. You should be able to take phi negative and psi negative and get the same result. So the CASP experiment, they sent out these sequences, and this is what David Baker's lab came back with. It's not exactly right, but you can see the helices are in the right place. So these are some of the very first blindly, correctly predicted protein folds, happened around the year 2000. This is the beginning of the Rosetta program. This is protein-protein docking. This is a very non-humble tour. This is some work that I did when I first came to Hopkins. This is another blind challenge where we're given two proteins and we have to dock them together. So the problems, the underlying biophysics is the same. We're using Rosetta, again, the same program you're using. And this is, the two colors is our prediction and the experimental result of the complex. And we can see that we've correctly predicted this orientation and we've even predicted these side chains in the right place. So again, this is more evidence that our strategies are starting to work. This is a very recent result from a couple people in my lab, Brian Weitzner and Daisuke Kuroda. Antibodies are hugely important to the pharmaceutical industry. It's about a third of the drug pipeline. A lot of times you develop antibodies by injecting stuff into mice, and they give you sequences that then they make. They don't necessarily know why they bind or how they bind, or if they want to manipulate them, how to make them bind tighter. So one important step is to get the structure. Structure isn't always easy to get experimentally, so we have computational methods to predict the structures. This is the H3 loop. This is the one that's most important for binding, and we get these predicted within an angstrom in some cases, not all cases. Again, this is a blind prediction where drug companies had antibodies, they gave us sequences, we predicted them, and then afterward they showed us the structures. These are selected results. There's a lot of results that don't come out right, and that's why it's still research. On the design side, the first designed protein was a zinc finger by, it was a zinc finger. This is a zinc finger fold, and usually there's a zinc right here in the middle. What Steve Mayo's group did with Basil de Hyatt is they pulled the zinc out, and they used the computer to fill up the space with side chains, notably these aromatics, and they got the protein to fold and be a well-behaved protein without the zinc. So that was the very first protein design by co-opting a natural protein and having it do something else. Skipping a few years where there were other milestones, but the first ab initio design fold, this is Brian Kuhlman using Rosetta in David Baker's lab, literally drew on the back of an envelope a bunch of strands in a helix in shapes that don't occur in nature, in a particular fold that doesn't occur in nature, digitized it in the computer, made a sequence, and it folded into that shape. So that was the very first ab initio, completely new fold in 2003, so 10 years ago now. The first enzyme was reported in 2005. We'll talk about that later. There were problems with it. In 2008, this is a Kemp elimination reaction where they position side chains to make this elimination reaction go. It's a pretty lousy enzyme compared to what nature does, but it is active. So there's work to be done on that. Small molecule binders, just last year, Chrissy Tinberg, again with Rosetta. This is she made a pocket, put a small molecule in there, and shaped the pocket by redesigning the side chains and stabilizing the fold around it, and she got micromolar binders out of the computer. A common strategy today is to get a starter out of the computer and then use evolution to evolve something stronger. So from that, they were able to get picomolar binders through experimental selection techniques. Nanotube binders. This is Gregorian working in Bill DeGrotto's lab. Here's a nanotube, and Gevorg looked at the geometry of the nanotube, put helices around the side, calculated the different ways helices could pack around it, and then designed side chains to pack. And what he can do is actually recognize different size nanotubes and different ways of wrapping the nanotubes up, wrapping the graphene sheet. So that came out a few years ago. And a couple years ago, protein cages. If you could do a protein interface, this is a trimer that was already occurring. They took the trimer and made an interface between two trimers at an angle such that you put a bunch of the trimers on an octahedron and you get a cube. This is Neil King's group, Neil King's lab. I've been using Rosetta, and there's now 2 component cages, and they're trying to figure out how to put things inside. So that's some of the exciting things that have been done in the last few years, and that's the kind of stuff that we want to learn about. And again, about half of these are done with Rosetta. There are other codes, but Rosetta will get us, we'll have access to all the routines that did this and talk about the methods that do that. Okay. I wanted to do geometry today, but I'm not sure I should start it now. I've just got a couple minutes. Are there any questions? Okay, we'll meet Monday in the computer lab. If you have a chance on your own computer, get PyMol, get PyRosetta. I've got homeworks here that I'll pass around.